Here is a claymation of humoral immunity for kids. Here a pathogen is introduced into the body, which is bacteria or blue stars. In the second stage, phagocytosis or digestion occurs. Here, the red lobster or antigen-presenting cell, B cell or macrophage, phagocytizes or digests the bacteria, blue stars. In the third stage, antigen presentation occurs. Here, the black square, which is the antigen, is presented to the MHC2 protein, a yellow square, on the cell's surface. The MHC2 protein then fuses with the antigen. In the fourth stage, helper T cell recognition and activation occurs. A helper T cell, a brown hand for paper, is recognized and activated to bind to the B cell, the purple rock. The helper T cell has T cell receptors that help recognize the antigen on the class II MHC protein. Here these cells bind to B cells to begin to reverse the immunity attack on the body, which I will talk about later. The fifth stage is B cell clonal selection. When the B cell, a purple rock, is stimulated, it clones and produces either memory B cells, purple rocks with spikes, or plasma cells. Most B cells clone and differentiate into plasma cells. B cells that are not stimulated do not get cloned. The sixth stage is plasma cell differentiation. Here, interleukin or chemical signals are secreted when the activated helper T cells bind with the B cells, and this stimulates the B cells to differentiate into plasma cells. Plasma cells are rocks with a loop on the top, and they are derived from B cells and prepare the cells for antibody production. The antibodies are seen later as googly eyes. The plasma cells synthesize and secrete these antibodies. Memory B cells do not produce antibodies. In the seventh stage, antibody production occurs. Here, googly eyes are antibodies that are produced. These antibodies bind to antigens, which are foreign proteins, and inactivate them. Each antibody binds to one kind of antigen. These antibodies can use 4 different ways to render antigens harmless. The first way is agglutination, where antibodies immobilize microbes and antigen molecules and prevent them from spreading through the tissues. A second way is through complement fixation, where antibodies bind to enemy cells and change shape, exposing their complement binding sites. Antibodies are a defense against such foreign cells as bacteria and mismatched erythrocytes. Third way is through precipitation. This is where antibodies bind to antigen molecules. These processes can be removed by immune clearance. And the fourth way would be through neutralization, where antibodies can neutralize an antigen by binding to these active regions and masking them. The final stage is immune clearance. Here, the macrophages destroy and clear out foreign antigens in the bloodstream and protect red blood cells, and this is shown through the brown trash can.